which is what it costs in the United States for a treatment.
Dr. James Allison and Dr. from MD Anderson Cancer Center and Dr. Takeshi Honjo from Kyoto University, they were awarded the Nobel Prize in Physiology and Medicine 2018 concerning their work on immune checkpoint blockade. Could you please explain how this cancer cell can escape the immune system? And what's the new strategy behind developing this drug and how to uncover this antigen and make them target for new therapies?
Yeah, so the idea is that PD-1 stands for programmed cell death receptor 1, and PD-L1 is programmed death ligand. And it turns out that in the immune system, there's a yin and a yang, or an accelerator and a brake, however you want to use the analogy. And if you get a vaccine, let's say against tetanus, you develop antibodies, and then eventually you have to turn off that response because you don't want your whole immune system to be doing nothing but developing antibodies to tetanus. And the same on the cellular side. So with a mumps vaccine, for example, you induce T-cell responses to the virus and or to viral infected cells. And same story, you can't just have it continuously look at that. But there's long-acting memory cells and so forth. So it turns out that tumor cells, and the ones that are the most interesting so far, are melanoma, and kidney cancer. But as you know, bladder cancer in terms of GU oncology also turns out to be this way. Tumor cells have sort of co-opted this turning it off approach, and they can make the PD ligand, PD-1 or PD-2 ligand, which will go to the T cell and kill the T cell. So the tumor cell that's the immune system that is trying to reject the tumor is suppressed by the tumor cell itself, making this protein that is a part of the normal yin and yang of the immune response. And the discovery was that if you could throw a monkey wrench into that by an antibody, a monoclonal antibody, either against the receptor, the PD-1 receptor, or the ligand, you could stop the tumor from suppressing the immune system and do that. So the most successful are in patients who have very high tumor mutation burden, or microsatellite instability, which is another indicator of high mutation rates, if you will. And if you use antibodies to block that, and one of the first was pembrolizumab, called Keytruda, of trials now in a variety of tumors. They're called Keynote 2, 3, 4, etc. Were done, and then all the other pharmaceutical companies jumped in and have developed other mAbs, so tislelizumab and many others. There are at least 5, I think, approved for bladder cancer. So inhibiting the PD-1 response. But yeah, so if we get back to GU oncology, the interesting thing in prostate cancer is that it's a so-called cold tumor. And even though there's a reasonable mutation burden, as I've already mentioned, if you just give the PD-1 inhibitor, like pembrolizumab, for example, to unselected prostate cancer patients, the response rates are fairly low. In melanoma, a third of them have a significant response or even a complete response. And the same in platinum-resistant bladder cancer patients, the response rates can be very high. But in prostate cancer, response rates are in the 5% range and so forth. So the current hot area of research is to try and make it a hot tumor instead of a cold tumor. Can you find a vaccine, for example, or some other mechanism for getting the immune cells to infiltrate the prostate cancer and then add the blockage drugs that inhibit the tumor's response to the infiltrating tumor attacking immune cells, NK cells, and so forth. So it's a great area of medical research, and some remarkable successes in melanoma in particular, but also kidney. And then it raises the question of, okay, if you have a dramatic response in a third of melanoma patients, why isn't it working in the other 2/3? And that's where the research is, headed. Can you figure out why it works in some patients and not others? And if you can, how can you make a patient a responder who might not respond otherwise?
Okay. Another aspect is bispecific T-cell engager, BITE. Could you explain this? How the mechanism it works? How can you create an antibody that will both engage the cell, the malignant cell, and the T cell?
Yeah.
Lymphocyte itself.
So it's another hot area of research. So antibodies are shaped like a Y, and this piece over here has a hypervariable region. Where my arms are is the constant region of the antibody, and then these are the two variable regions. So with molecular biology, actually make one of these bind one antigen and one over here bind a different antigen. So you can make this one bind a T cell and this one bind a tumor, and then maybe bring the T cells together, the killer T cell next to the tumor cell. But it's even more complicated, because if you combine some of that technology with things that will also stimulate the surface of the T cell with the HLA locus and all that, you can have it not only bring the target and the killer into proximity, but also stimulate the T cell to become a more effective killer cell. And then the BITE part is you can get rid of all this FC portion of the antibody and make some artificial ways of binding those together. So you just have something that looks like that. And it's a much smaller molecule, and then figuring out the half-life of those molecules. It's a very rapidly moving area, and I'm certainly no expert, but as you pointed out in the blog, I tried to explain it a little bit with some simplified drawings that I could understand from the literature.
So that's what we need at our level.
Yeah, no, clearly needs somebody
to be a PhD
make it easy
in immunology.
Yeah,
understand the whole
or molecular biology, or both, right?
So any current indication of this therapy?
I think that the most successes so far with this type of the BITE type treatment have been in the liquid tumors. So lymphomas and leukemias have been the place where there's been the most success. And I think getting it to work in solid tumors has been challenging, but there's a lot of ongoing research and efforts to make it work.
PSMA antigen targeted monoclonal antibody. How can we combine this PSMA with this radio-labeled constructs, especially 177 lutetium? What is the principle?
Yeah, so
what we call now theranostic. What is theranostic?
So PSMA, prostate-specific membrane antigen, is a protein on the surface of cells. It's also on endothelial cells and neovasculature and so forth, but it provides a relatively prostate-specific target. You can target that molecule on the surface of the cell either with a ligand that binds to it, and that's where the PSMA scanning that we talked about is mostly using ligands that will bind to that, and then you hook a radioactive positron emitting isotope to that, like gallium-67 or whatever. The other approach would be to make an antibody against that. Either way, with a ligand, you can deliver radioactivity, and the radioactivity could be a PET active agent, or it could be an agent that emits electrons or alpha particles or whatever. So the lutetium-177 story is lutetium-177 hooked to either I think J591 is the antibody, and there's some research going in that direction, or one of the ligands. And now you are delivering radioactivity that could either be used, you talked about theranostic, let's say you use a low intensity radiation, if you will, that can be picked up by a scanner and not hurt the patient and not have any issues. Or you can make a very hot isotope, Lu-177, in much more higher doses, if you will, and that will deliver a therapeutic. So when you get into the higher dose range, then you get into the therapeutic index. So how much radioactivity really goes to the tumor versus is soaked up by the liver, for example. Yeah. You don't worry so much about that with an isotope that is being used for diagnostic purposes, but it certainly limits, if the isotope is secreted in the urine, it can be toxic to the kidneys, or if it goes to the liver, it can be toxic to the liver. But the general idea is try to find something that is bound to the tumor cells, has enough radioactivity to either detect or in higher doses to cure or kill tumor cells. And Lu-177 PSMA is an exciting area of research, and I anticipate, I mean, the trials so far look very promising in patients, and for prostate cancer, the castrate-resistant patients, I think we'll probably see approval of one of these agents within the next year or two in the United States, at least.
Right. It can be combined with other immunotherapy, this principle of BCMA.
Well, I don't know. I think that's in theory, yes. And then you get into the issue of, yeah, if cancer cells are dying, they're releasing antigens, and you can get what's called antigen spread. And so if you were using an available immune approach, and these immune approaches can be combined. For example, we talked about Sipuleucel T. Why not combine that with a PD-1 inhibitor? Yeah. And then if you wanted to get really fancy, why not combine it with something that was killing the tumor cell with radiation, either external beam or an isotope, Lu-177. But the complexities mount as you add each layer of treatment, and the toxicities.
Yeah, it's going up.
It becomes a real challenge. So I think it's exciting, but a long ways to go to figure out the right sequence and the right combinations.
Let's move to genome editing. This is a group of technology that gives scientists ability to change organism DNA, and this technology allows material to be added, removed, or altered in a particular location of the genome. One of the most important technology is CRISPR-Cas9. Has generated a lot of excitement in the scientific community because of it is faster, cheaper, accurate, and more efficient than other existing genome editing tools. Tell us about the potential of CRISPR-Cas technology in cancer, immunotherapy, and beyond that. What is the next in research in this field?
Yeah, well, you're getting
beyond
you're getting beyond my ability.
I'm abusing your kindness.
Well, so the idea is that you can guide enzymes to the DNA and clip out parts of the DNA and insert other parts of the DNA with the CRISPR-Cas9 system. So you can very specifically even change a single nucleotide mutation, for example, in sickle cell, we'll say.
Yeah.
So you can theoretically and in practically take a patient who has
this is one single gene, single disease that lead to one.
Yeah,
single gene.
A single nucleotide. You can replace that nucleotide with the correct one, and then if you do that in stem cells, you could expand the stem cells and then re-inject those into a patient who has like a bone marrow transplant. And then if you've done it correctly and you've done it with the patient's own cells, then the patient might have sickle trait instead of sickle disease. unless you can replace all their stem cells. So single gene mutation things are the target for this, and there have been some successes already using that type of editing. The challenges, as I understand it, are off target. So you can also occasionally have the enzyme, the CRISPR-Cas9 system can insert something in or change a gene somewhere else that you're not targeting. And there are papers coming out all the time about increasing the fidelity of targeting, so that it's getting, as I understand it, getting better and better at just hitting the gene that you want to change and not other places in the genome that could be dangerous down the road. It's moving very fast, and in the long run, it could be very exciting. One of the things, George Church, who's at MIT, is one of the leaders in the field, and if you read some of his articles and stuff, there's just amazing things that can be done. And so he's got a collaboration with a guy in Siberia, and they're trying to figure out if they could do enough mutations in elephant genes to bring back the mastodon. So, mammal, yeah, the mammoth, yeah. So it gets pretty wild as to what can be done. But practically speaking, there have been a few successes in human monogenic diseases.
Yes.
And there's a retinal disease that can be corrected, and so forth. So it's a fast-moving field and over my head in terms of actually practically doing it. The ethics of it have been highlighted with the Chinese scientist who treated the twins with altering the genome so that you wouldn't have HIV infection and so forth. But all those off-target issues were not well worked out, and at the present time, changing the germline is not ethical and being challenged.
Germline may be handed down to the next generation because it will be inheritable.
Right.
Right.
Yeah. Permanently. Yeah. Now the exciting part of that. Well, and it's particularly true. I know that some of your audience is Africa, but you can do a thing called gene drive with this approach, and I'd have to review it to actually explain it well. But you can alter a mosquito so that it's passed down to the next mosquito, and the mosquitoes become sterile.
Yeah,
and then you can potentially eliminate an entire species of mosquitoes. Yeah. And that's on. In theory, it's possible, and it could be a good thing to do. But you start getting into, okay, when you affect the whole biome that way, maybe those mosquitoes have an important role in feeding fish.
You're messing up with nature.
Yeah, you're messing with Mother Nature, right?
Yeah. Great. We'll go back again to the prostate 8 blogs, in which you try to sift through a tremendous amount of article to give a take-home message to your reader. You just remind here that you are receiving hundreds of notification of targeted posts that fill your mailbox each day. And your posts you target an audience of experts without ignoring and confusing the layman. So this requires, of course, to strike a balance between neither going too much into the detail or skipping over what is really important and fundamental. How the idea came about and how often you write in your page, and is it regular or just on demand when something new pop up?
So it's been a fun project. So I had an interest in the internet kinds of things, and in particular when I was working with the ASCO Online project, the early stages were kind of the email stage, if you will, and then blogs became available, and I can't remember the name of the company. A company asked me if I would write a blog. I think it was on prostate cancer, maybe in cancer in general. But so I did that for a year or so, and then they sold the company to another company And in essence, we're just taking advantage of, if you will, scientists who are writing blogs in order to commercialize it. So they got advertisers and so forth. So I stepped away from that as a commercial thing. They may have paid me a little bit at the time, but anyway, it didn't seem like something I wanted to continue. So then by that time, this is probably 20 years ago, 15, you could start having your own blog. So for $99 a year at, I think it's Word something or other, you can buy space and have a blog. So I started doing that, and as you pointed out, my focus, my target group is my patients. So what happened was, as some of these new technologies and prostate cancer clinical trials became aware, so there'd be an article in the Wall Street Journal or the New York Times or something, and a patient would come in and or send me an email and say, look at this article, what do you think about this? What about this new Provenge or Sipuleucel T, or what's going on with caspase and all of that. And if it was a popular article, sometimes you'd have 4 or 5 patients asking you. Well, to sit down and have this kind of an interview with each patient takes a long time. So I thought maybe I'll just write a blog if there's something that looks, hot, and then encourage my patients to go there. And the nice thing is you can do a hypertext link within the blog, so that if people are really interested, they can click on it and they can go to the actual scientific paper. So I do what I hope is comprehensible to a layman. Here's the story of PD-1 inhibitors. Yeah. But if you really want to read about it, here's a review article in Science. You can go, review that.
That's for the experts.
Of course, yeah, and many of the articles are only available if you have a subscription to the magazine or access to an academic library, for example. So I try to find sources that the patients can get to that are valid. So Wikipedia being a great example. You can often find a really nice review that's well written in Wikipedia that again has references to very deep scientific information. Anyway, so patients I see a patient, they suggest a topic for a blog, or I go to the ASCO GU meeting and I see a new clinical trial that is, to me, a very interesting one to keep an eye on. And then I write a blog about it. I try to do about one a month. I think you can sign up for being notified when I post a blog. So I think there have been about 600 people sign up for it. So I have a following.
Yeah, your star.
It's mostly through my patients. It's mostly written for my patients, and it's been a fun project because, like you said, I have, and you do too, I'm sure. You have dozens of emails every day with prostate stuff in it, and if you
cannot read through all this,
you can't keep up to it. So this is sort of a way of trying to help patients keep up a little bit.
Okay, Michael, thanks for bringing us up to date on the latest in immunology field, and will be certainly very exciting moment ahead. So we can't wait to learn about this new discovery in this field. It was a pleasure talking to you, Michael. That's very kind of you for taking all these questions and bearing with us all this time.
Well, Zen, it's so wonderful to see you again. I guess it took the COVID epidemic to bring us together and figure out how to use Zoom. I think I had used Zoom once before 3 months ago, and now it seems like there's a Zoom meeting every day.
On the regular basis.
Yeah, this is my second one today, so here we are. But I look forward to COVID going away and being able to visit you again. It was
that will be a pleasure to have you here in Morocco.
Yeah.
All the best, Michael.
All right. All the best to you as well.
All right. See you later.